Hello, everyone. We're going to start chapter 11 today, which is going to talk about Mendelian genetics and how your genes affect how your physical characteristics are. This is Gregory Mendel. We're going to talk a lot about Gregory Mendel in this first section, but let's just jump right into it. Remember, anything on this black background you don't have to write down, but I like to go over this because it's really important for you to understand kind of what happened before Mendel and during Mendel. But I'm not going to really dive deep on assessments about this. I want you to know the concepts of genetics, not really the history. Okay, so Mendel here. Before Mendel, we thought that the blending hypothesis traits blended. You can see here this yellow and the blue. In this case, when they blend, they're going to create green. Now, we do have some evidence that supports this, but again, we're going to see it's actually incorrect. The blending hypothesis was disproven by Mendel back in the 19th century. To give you an example, what this blending hypothesis is stating is you can see this red and white flower. If they would mix, you would get pink. Even if you start out with all red and white flowers, eventually you would get all pink flowers because they would just keep mixing and mixing and mixing. And obviously, we don't see that today. We see that traits are independently inherited. So this blending hypothesis is wrong. But Gregor Mendel was an Austrian monk you can see here during the 1850s, 1860s. He did study math and science at the University of Vienna, which means he was educated. He just wasn't like a person that didn't know about science. He was also actually a substitute natural science teacher at a local high school, so I actually have a little bit of like connection with him. I think it's a really cool. He was a monk, but he was also a natural science teacher. So he did this a really important experiment within genetics on pea plants. We're going to talk about those pea plants here in a little bit. But he chose pea plants because they're short generation time, so it means that they can reproduce very quickly, and they have very distinctive phenotypes. He worked with these pea plants for over 8 years and collected 28,000 pea plants and their data. So he had a lot of data, which was really crazy for the time. Science wasn't really big and well known. I would say big, but it wasn't really established as it is today. And a lot of his procedures, we'll say, were really accurate. There are some debates within deep scientific communities whether his data was maybe fudged a little bit because it's really accurate for a monk that's in Austria, present day Czechoslovakia at the time. Like they're like, how could he have been so accurate? But he maybe manipulate a little bit of his data doesn't mean that it's not correct. I mean, we could do these experiments and get the same data now today. You can see his garden back in the 1800s and his garden today. Very beautiful. As you can see here, with the peas have a lot of different phenotypes, and these are physical characteristics we're going to talk about next. And you can see they're very distinctive too. They're not very common between each other. You can see like the white and the purple flowers either having tall or short. So these are very distinctive. So what he did was he started to breed the same plants together, so like tall plants and short plants, and he called these true breeding. And what happened was he got these two true breeding plants after a while. So he had all these tall pea plants and these short pea plants, and he crossed them. He crossed two of them, and what he predicted was he should get medium, right? Because if the blending hypothesis was correct, these two are going to blend to make medium, and then if you cross two mediums, you're going to get all medium. Now, he probably started thinking about this beforehand and started thinking like, well, I'm not seeing any of these medium plants, so this blending hypothesis probably isn't true. And this is called a monohybrid cross. You're looking at one trait here. What actually happened was he got all tall plants, which really confused him. He's like, that shouldn't happen. If the blending hypothesis is true, we should get medium plants. So what he did was he did some little bit of self-fertilization, and yes, a little bit of influence, but they're plants. That's not as, I don't want to say important, but they can happen. And he got 3-to-1 ratio of tall to short. So this short pea plant kind of reestablished itself in this next generation. This was odd. This was big news here, and Mendel knew that something was going on here. Interestingly, some people are like, "How did I've been asked, how did Mendel know which plants bred with which plants?" And again, you wouldn't think it, but pea plants reproduce sexually. I mean, they have the male and the female parts. You can see here the pollen. It's going to fertilize the carpel here of another pea plant. So, and he actually used these. I think they're like paintbrushes. We could do this in lab, but again, on this year with COVID, we're not going to. But you can actually transfer that pollen and make sure you know which ones are which pea plants are mating with which pea plants. I know it sounds really weird that plants are sexually mating, but yeah, it happens. So just a little bit of context here with the naming here. P generation, that's the original generation. That's your parent generation. F1 is your first generation. That's the offspring of the P generation. And F2 is the offspring of the F1. And you can see here we go on and go forth. So like if your parents are the P generation, you are the F1 and your kids will be the F2. So you're going to see this terminology throughout this chapter, and maybe even in some, like I know Mr. Sharp talks about how this terminology is used in like magazines for selling farm like seeds and cattle and things like that. They have different ways they name those things. So this is his actual data, and the really cool thing is he got really close to all of his numbers between with a 3 and a 1. So again, this is the F2 generations. We have the tall to short, yellow to green, purple to white, axial to terminal, and this is his actual data. You can see here he got really close to that 3 to 1 ratio almost every single time. Now you might think, hey, Mr. Fradoff, like why is this not exactly 3 to 1? Remember, if you flip a coin 100 times, you're not going to get 50 heads and 50 tails. You might, but it's kind of improbable. I don't say improbable, but the more likely is that you get like 52 and 48. It's very hard to get those perfect numbers in nature. And again, these are thousands of points of data, but even in nature, these things don't perfectly work out. So a couple more terminology that we got to know. Genotype. This refers to the alleles that an individual has. So you can see the genotypes here. Each one of these is called an allele. We talked about these a little bit last chapter, but your genotypes, I always say, they are 2 letters. Allele, genotypes are 2 letters, and alleles are 1 letter. Phenotype is your physical characteristics. So this is like the proteins and how they are aligned and formed and structured to make who you are. So your genotypes code for your phenotype. 2 other words we're going to learn here is homozygous and heterozygous. Homozygous are the genotypes with the same alleles. Heterozygous are genotypes with different alleles. So you can see homozygous, we have 2 of the same alleles, and then heterozygous, we have 2 different alleles. A couple more terminology that we need to know, or a couple more vocab words. Dominant allele is a capital letter, and that is DNA producing fully functioning proteins. Recessive alleles don't produce a protein that's functional, or it's like semi-functional. It doesn't really produce what the dominant allele would be. This is different than what you learned in your 9th and 10th grade biology class. So we're kind of taking the next step here already. This is not just like a mass trait. It is a improperly produced protein. So that's why its mass is because it's not going to be producing the right, or I should say the dominant protein. Okay, so at this point, what we're going to do, and I can't do it here on my laptop, but we're going to just talk about it. In the first crossing, he had the tall and the short. You would put big B, and little b here. So again, big B for the one parent, and little b. You're going to ask, hey, Mr. Froth, what is there a difference between putting the little b's and big B's over here? Not really. There's really no difference because you're going to get everything the same here. In each one of these, you're going to get a big B, little b. So that means that all the resulting offspring in the F1 generation are going to be tall. So you could fill this out again. The genotype, this is for the offspring. You're going to get 0, 4, and 0, and 100% are going to be tall. Oh, I do it a little bit there. Oh, I do a lot there. Here we go. In the F2 generation, you're going to cross two tall plants, and you're going to get that big B, little b, big B, little b. You can see here we're going to get this big B, big B, big B, little b, big B, little b, little b, little b. The ratios work out to be 1 to 2 to 1, but the phenotypes are 3 tall, what we saw, and 1 short, again, what he saw. You can see here where he crossed. Now I'm not using the tall and short, I'm using the yellow and green here. Excuse me, sorry guys. He's going to get all, when he crosses these, he's going to get all yellow. So that means yellow is a dominant phenotype here. When he crosses 2 of these, he's going to get 75% or 3 to 1 ratio of yellow to green. This is just showing you the genotypes here, the big Y, and little y, little y. You get 100% big Y, little y, and then you get that 3 to 1 ratio of yellow to green. Not sure if heterozygous or homozygous dominant. Yeah, because both are going to show that are the dominant phenotype. All right, so 2 other words. Again, we talked about alleles a little bit ago, but the locus is the location of a gene on homologous pairs of chromosomes. We're going to see these really come in importance later on in the next couple sections. And alleles, again, just alternate version of genes. You can have like the yellow pea pod or the green pea pod here. All right, the law of dominant states is going to state that only one form of the trait will appear in the next generation. So we can see here, this tall trait is going to be what is expressed in the next generation. However, even though we're crossing two of these F1 generation pea plants, you will have this short pea plant be produced because there is no dominant gene there. You're only going to have the recessive genes being expressed there, which is the short pea plant. If an organism inherits at least one dominant factor, they will exhibit the dominant trait. If an organism inherits two recessive factors, they'll exhibit the recessive trait, just like I said. Law of segregation. Each organism has two factors for each unit. Factors separate during meiosis. Remember, what we're actually doing here is the meiosis part. So like these up here are the sperm and these down here are the egg, or vice versa. They're just showing you the chance of the offspring having one of those two alleles. They're going to segregate independently of each other. So you can see here that where the F1 gametes had a big Y and a little y, they're going to separate. You're not going to get both from one parent because, again, you only, with the, let's see, I can get my picture up here. With the, like, if you have two homologous chromosomes, you're either going to get this chromosome or this chromosome. Now, there will be crossing over that occurs between them, but we're going to talk about that later on. Remember, you only get one of two chromosomes when you, after meiosis and fertilization. Ooh, let me go back. So, interestingly enough, Mendel proposed a particulate theory, which he said these traits aren't blending; they're being inherited independently of each other, and he didn't know what it was. So he called them particles. So he said, "Hey, they're inheriting something. We don't know what they are. They're being inherited, and what those particles became, and we figured out later on, but it wasn't for about 50 years, were genes and chromosomes." It wasn't until like 1902, 1903 that Bavaria Sutton are two scientists who were working with fruit flies, we'll talk about later on. And they started noticing that chromosomes were being inherited from the sperm and egg. And that's when they were like, hey, I think I remember reading about this in, I think it was college. And they read some of Mendel's work and they said, hey, this guy was actually really accurate. Let's reread his stuff. And they figured out, like, holy crap, this guy was really, really accurate. And so again, those alleles, this is such an important, oh my gosh, this is so important here. Big A, little a. Those are alleles that are at different, I should say, they are alleles on chromosomes. They are the same genes, but different alleles. You can see here, you're going to independently inherit one of these or one of these. So again, you're going to pass on one chromosome from your parents to your offspring. I hope that makes sense. This is really important for you to understand. These alleles here, which is a genotype, they're going to be inherited from your parents. So again, you got one from dad, one from mom, but they come from chromosomes. Ooh, let's stop there.